in this video I am going to continue your protein topic. Correct? This is related with Unit-14 biomolecules. Then today's first topic is isolation of proteins. Correct? That means separation. So very easy. Proteins are extracted from crushed tissues of seeds of plants and animals by water. Correct? So seeds of plants and animals by water, 10% NaCl, 2.0%, 0.2% KOH, and 70 to 80% ethyl alcohol. Correct? By water, 10% NaCl, 0.2% KOH, and 70 to 80% ethyl alcohol. Proteins are obtained from different extracts either through dialysis or are precipitated by any appropriate reagents like ammonium sulfate NH4 whole twice SO4, MgSO4 and alcohol. Correct? So in this way isolation of proteins takes place. Then I am going to take characteristics of protein. So proteins are generally colorless, odorless, tasteless, amorphous solids. Correct? Colorless, odorless, tasteless, and amorphous solid substances called proteins. And proteins can form salt with acids and bases both due to the presence of single bond NH2 and single bond COOH, that means acid and base group both. And protein get hydrolyzed into constituents amino acids through acids, bases or enzymes. On hydrolysis, proteins gives proteoses, then proteoses on hydrolysis gives peptone, and peptone on hydrolysis gives peptides, and finally peptide on hydrolysis gives amino acids. So on oxidation, on hydrolysis they hydrolyze into constituent amino acids, and they get oxidized on combustion or putrefaction and form substances such as nitrogen, carbon dioxide, H2O, and amines. And also remember students, the foul odor produced on decay of any dead body due to the formation of amine. Actually, proteins are odorless substances, but on decay they form amines. So due to formation of amines, foul odors is produced. Correct? So basic reason is formation of amine. And next property is coagulation, also called denaturation of proteins. Students, on changing the temperature or pH, the secondary and tertiary structure of proteins are disturbed. Correct? Due to change in temperature and pH, secondary and tertiary structure of proteins are disturbed, and they are not able to perform their normal biological function. This is known as denaturation of protein. Correct? This type of coagulation is called denaturation of protein. For example, when egg is boiled, it's a water-soluble globular protein. That means albumin present in white part of egg is coagulated, and it converts into insoluble fibrous proteins which coagulate. Correct? So this is called coagulation, and the process is called denaturation of protein. And then I'm going to discuss test for the detection of proteins. Many tests have been used for the detection of proteins. Some important tests are xanthoprotein test, biurate test, Milon's test, Mollis test, Hopkins cold test, Menhydrin test, nitrocrucide test, lead sulfide test, and precipitation test. Now I am going to discuss some functions of proteins, and the first function is formation of new tissues and their repairing within the body is the main function of protein. And some proteins functions as enzyme, which catalyzes all biological reactions, and they also produce energy in absence of carbohydrates and fats. Correct? And control the motion of muscles, etc. works are performed by protein. And then I'm going to take disease due to the deficiency of proteins. So due to deficiency of protein, basic disease is called kwashiorkor, which has the following symptoms: swelling in body, skin becomes dry and decolorized, activity of liver decreases, and this causes loose motion. Correct? Now next is uses of proteins. So some main uses are protein as an important constituent of food, such as egg, meat, pulse, etc. in form of eggs, meat, pulse, etc. And insulin as a protein hormone regulates blood sugar in the body. So it is very important protein for sugar patients. Casein is used in making artificial wool and threads. Casein plastic is used in making buttons. And gelatin is used in making capsule, photographic films, and plates. Blood protein, thrombin and fibrinogen take part in the clotting of blood. So proteins are very useful for us. Students, this is our today's topic. I think this is enough for this video. We will take next topics as in next videos. Okay, goodbye.